Nobel Prize really has to do with not just a really important discovery, but it has to do with the impact that that discovery has in the field. And if you look at it, I mean, the discovery, well, let's back up for a second. His structural elucidation of THC was extremely important scientifically and extremely important in the chemical literature, but that wasn't really worthy of a Nobel Prize because, other people had isolated it first and other people had talked about its structure, but then Raffi kind of came in and cleaned up all the previous work. But what certainly was notable was his work that they did after the discovery of THC and after they found out that it was the principal active component, so-called toxic component, or the thing that led to intoxication from cannabis, was how does it work?
We must work untiringly so that our children are obliged to learn the truth. Because it is only through knowledge that we can safely protect them.
Curious About Cannabis is dedicated to providing reliable cannabis science education to anyone curious enough to learn. To get access to free courses and other educational resources, visit learn.cacpodcast.com and become a Curious About Cannabis member for free. The Curious About Cannabis book provides an incredible crash course in cannabis science through over 500 pages of content filled with photos, activities, science experiments, games, and more to help guide you through your personalized cannabis education journey. This book has become a trusted textbook in colleges and universities across North America and is absolutely perfect for serious learners, as well as cannabis educators, bud tenders, clinicians, patients, and caregivers. And special thanks to the many individuals, companies, and organizations that have helped Curious About Cannabis meet our mission of becoming the #1 trusted source of cannabis science education on the planet. This includes organizations like Credo Science with Ethan Russo, The Kenigma, Treadwell Farms, The Spellman Report with Kevin Spellman, The Workshop, Green Earth Medicinals, CBD National, Magnolia Botanicals, and more. Visit cacpodcast.com/sponsors to learn about our sponsors and go show them some love for helping us spread cannabis science education far and wide to anyone curious enough to learn. If you like Curious About Cannabis, consider checking out some of these other learning initiatives by Natural Learning Enterprises.
Life, mysterious and ephemeral, breathtaking, yet fierce and unyielding, grounding, yet transcendent. It's a curious thing. Let's explore it together. Isn't Life Curious? Available at isn'tlifecurious.com or wherever you experience podcasts.
And now, back to the show. Hey everybody, this is Jason with Curious About Cannabis. Thanks so much for tuning in once again. So this is a very special episode. I'm here with my friend Mark Scialdone, and we wanted to do a bit of a kind of a tribute to Raphael Mechoulam's life. As many of you know, it's been a little over a month or so now. Raphael Mechoulam passed away. And one thing that Mark and I have in common is kind of a fascination with the story around cannabis science research, cannabinoid research, and obviously Raphael Mechoulam was a huge part of that story. And so we wanted to take time today over the next hour and a half or so to really just unpack, because I think a lot of people haven't been exposed to the full story. They hear little tidbits about what Raphael Mechoulam did and contributed to, but I don't think they've really been exposed to sort of the kind of grander story and where his work fits in that grander story. So I'm hoping that we can do it justice today. Mark, thanks so much for being willing to join me and try to take on this endeavor. It's a big story to tell.
It sure is.
Let's go all the way back. Yeah, well, first of all, I'll say to anyone listening to this, we have a sort of a deep dive on cannabinoid science history you can check out that Mark's already done. It's about 3 hours long. It's not focused on Raphael Mechoulam by any means, but it's if you're interested in sort of more details around some of the story we're going to be talking about, I recommend listening to that. It's really, really good. But let's kind of go back. I think, really the beginning of modern cannabinoid research as we think of it today, systematized, research, really started to take off in like the mid 1800s or so, in terms of techniques and things being applied that are in any way reminiscent of what we do today. So let's start there. What can you tell us about, the mid to late 1800s and what sort of set the stage for all of the research that would take place in the 1900s that Rafi was a part of?
Yeah, well, I mean, I think a lot of it stemmed from a publication that was published by uh, Sir William Brooke O'Shaughnessy, who was a medical doctor in England and was investigating the use of what they called Indian hemp
Right.
for its medicinal properties. And it was interesting. He in this paper, I think it was published in like the 1840s. That's right around the time when I think people started thinking that there was real medicinal properties to cannabis. And so I
Yeah.
think it's been used for a long time by several cultures, and I guess banned by other cultures
Right.
because they didn't like the state of mind that
Yeah.
cannabis.
Prohibition is not new. Yeah.
Right, but like I think I want to say that maybe it was the Greeks that were involved, but there's a Jason, you probably know this excellent YouTube video by Chris Rice on the lost history of cannabis, which talks about cannabis use in pre-recorded history as well as just in ancient history. And there were several cultures in the East, most certainly China and some of the other Asian countries where cannabis was used for not only its medicinal properties, but certainly for material properties, for
Right.
fiber, food, seed oil. So there had been, quite a bit of use of cannabis for a long period of time, but it hadn't really been studied medically, and really medical sciences in the early to mid 1800s were in their infancy,
Yeah.
or at least modern medicine. I guess you'd call it modern medicine.
That's how I like to preface it. Just like sort of as we think of modern medicine today.
Right.
Yeah.
Right. But anecdotally, I think there was stories handed down over the years, and
Hmm.
Mechoulam talks a little bit about this in the movie The Scientist, which again is another excellent video on YouTube that chronicles Raphael's career and contributions in cannabis. But I think that like when people started specifically needing it for particular medical conditions, the one story that he talks about was an ancient story that I think dated back to one of the early civilizations. I want to say Mesopotamia, but don't quote me on that. It was early civilization, maybe it was in Persia, I
Might
think.
have been the Scythians or the Persians. Yeah.
Yeah, and I think there was a king who had epilepsy, or some royal member of the royal family who had epilepsy, and they knew that cannabis was effective in treating that. They didn't know why,
Yeah.
and they knew that it had to be taken every day or constantly to treat the disease. But I think that's when people started really. I think when they read O'Shaughnessy's paper and talked about treating different maladies with cannabis, it started gaining popularity in the mid to late 1800s in the UK. And there's a famous story about Queen Victoria, who used cannabis to control menstrual cramps and menstrual pain, and other, things like that. I mean, if you read O'Shaughnessy's paper, he talks about giving doses of cannabis extract to dogs too, and looking at, whether it puts them to sleep or makes them happy or stuff like that. So that paper was really enlightening to a lot of people, because he actually took extracts and in the best way possible in the mid 1800s was able to measure some type of cause and effect of the extract itself. Again, this is before molecular sciences. So
Exactly.
They had no clue what was in there. And so later on in that century, in the 1800s, people started investigating, could we actually isolate this? And typically in a extract or what was really called a tincture. Tincture typically was an alcoholic extract where you just boil off the alcohol, and then what's left is kind of like everything that's alcohol soluble. And so alcoholic extracts were quite common. And there were some early investigations that looked at, geez, what is this that's in there that's causing this? But it wasn't till the pioneering work. We talk about this in the deep dive. This is a very important finding, which was the group in 1896 at Cambridge who first vacuum distilled cannabis extracts that they obtained from extracting charas. So we all know charas is a traditional form of cannabis that is just the sticky exudate. I think you can almost think of it like finger hash, or just the way people would just sort of wipe the oil off the plant with their hands and then kind of roll it together in a little ball. And so those kinds of preparations are probably high in cannabinoid acids, because it's the cannabinoid acids that are made by the plant. But then when they go to vacuum distill it, obviously it's going to be something that's going to thermally decarboxylate. So what they're going to get is a fraction out of that distillation is going to be completely decarboxylated. And I have a particular perspective on that work, because when I was making cannabis distillate and I measured the boiling point, the boiling point is something that's often misrepresented in the lay literature, right? We've discussed this before, right, Jason? So when Mechoulam first isolated THC in the Delta 9 form and then reported the boiling point, that boiling point was under a vacuum pressure of, I think, 0.05 torr, or 0.05 millimeters of mercury, which describes the depth of the vacuum. So when you distill something under vacuum, it distills at a much lower temperature than it would distill if it were just at regular atmospheric pressure. And so often the THC boiling point gets reported as the boiling point that Mechoulam reported in 1964 in his landmark paper, but they forget to mention that it's under a vacuum pressure of 0.05 torr. So if you try heating cannabis extract up to 155, not much is going to distill.
Right.
You have to put it under that vacuum. That vacuum, again, lowers the boiling point, because you're decreasing the energy needed to transition a vapor into a liquid into the vapor phase. So I believe that Wood, Spivey, and Easterfield, even though they report it as cannabinol, canna from cannabis, and all, because they knew that there was a hydroxyl group. So there was no descriptive analytical method that they could use. They just had to kind of take observations based on how the molecules behaved,
Yep.
and they could assign functional groups. And when they called it cannabinol, we know cannabinol is CBN, which is the fully aromatized derivative of THC. They were confounded. The earlier researchers were confounded by THC's metastability. So when I call it metastable, that means that it can be stable in certain forms, but in other ways or other conditions, it can rearrange or decompose or change its molecular form. And the earlier researchers were confounded by this because, as we know, there's a single double bond in tetrahydrocannabinol, and that single double bond, whether it's between the 8 and the 9 carbon or the 9 and the 10 carbon or the 10 and 11, these are all just structural isomers of one another. And if given the opportunity, the energetics of that landscape is you could scramble that double bond and put it in any number of positions. And this is what we hear about when we talk about delta 8 THC versus delta 9, which is the naturally configured material. As it turns out, delta 8 is thermodynamically preferred over the delta 9 configuration. So if given the opportunity, delta 9, which is what the plant makes, which is naturally occurring, will isomerize to delta 8. So for that reason, I believe that the plant makes no delta 8 THC. It makes no delta 8 configured cannabinoids. And any delta 8 which is found in analyzing cannabis extracts, I believe, this is my belief, is an artifact of the sampling method used that is promoting isomerization to the more thermodynamically favored. That could just be heating the sample. It could be shaking the sample. There could be trace amounts of acid that can do that. But a slight amount of something acidic as an impurity could cause isomerization of the naturally occurring delta 9 to go to delta 8. And it wasn't till Robert, so the isolation of the extracts was done by Wood, Spivey, and Easterfield, reported in 1896. They had a follow-up paper 4 years later trying to, again, get some more identification around the active ingredient. And what they did back then, Jason, because they didn't have NMR spectrometers and HPLCs and mass specs and stuff like that. So what they often did is they derivatized compounds to make them into a known compound. And if you could trace it into a known compound, you could kind of figure out what the unknown was. And a typical reaction would be something called nitration, where you boil something with nitric acid and you basically start decorating the molecule with these nitro groups. Well, the one thing that does sometimes is it makes a crystalline derivative. And so you can imagine that if you're working with a very sticky goo and all of a sudden you treat it with some kind of reaction conditions like nitric acid, and now all of a sudden you get a crystalline solid that you could filter and take a melting point. Well, you could try to figure out what the starting material is based on solving what the structure was. So a fellow by the name of Robert Sidney Kahn did a very, very important series of papers in the early 1930s, so about 30 years after the Wood, Spivey, and Easterfield paper was out there. And there was some other researchers looking at cannabinoids or trying to figure out what the active ingredients in cannabis were. There were some German groups that looked at it before World War II. But a lot of what we know about by just looking at the literature is that Robert Sidney Kahn, in his first paper in 1930, looked at the Wood, Spivey, and Easterfield data and said, well, I think maybe it's not cannabinol. He was the first one to propose the structure. Actually, the structure of cannabinol was not fully known, not fully proposed, but
Yeah.
he kind of sort of put it together based on some of the degradation products that they observed, had some theories about how these went together. And in 4 back-to-back papers, 1930, 1931, 1932, and 1933, he basically figured out what the molecular formula. He figured out the molecular formula for CBN, which was based on the Wood-Spivey and Easterfield data, but also figured that there's a tetrahydro compound that's converting over to CBN, and that's confounding the analysis to obscure actually what is the active ingredient. When they first thought it was cannabinol, one of the reasons why they were able to isolate cannabinol is because when you make the acetate of cannabinol, it's a crystalline solid, right?
Yeah.
So they isolated that, but it was in really, really low yield. And I think that yield is what tipped off Robert Sidney Kahn to think, you know what?
No, there should be more here.
There's other things in there, right? And so if you look at Kahn's papers, and it's just wonderful to just look at each one of those 4 papers, and you can see his evolution of thought to get him to where he finally, for the first time, identifies the structure and the formula of THC. And I think might be the first one to actually use the name tetrahydrocannabinol. So then after Kahn, 2 very important groups in very different areas. There was Alexander Todd in the UK, and then there was Roger Adams in Champaign-Urbana, the University of Illinois. And so this set the stage for Mechoulam to come in, because if you look at the papers that Todd and Adams published, this was just before the beginning of World War II. So you can imagine that, things over in Europe, my God,
right, exactly. Things are crazy.
Go off the hook. But this very, very important work was being done. And then when you look at Adams's work, so Roger Adams, the noted organic chemistry professor.
I was going to say, it's worth noting, like, Roger Adams is a big deal in organic chemistry.
Huge.
Yeah.
He's huge because, he had many graduate students and postdocs who ended up becoming professors at universities. So my generation of PhD chemists and graduate students, we were educated by the students who worked directly for Roger Adams. So it's amazing. I think he's my chemical great grandfather through my lineage. But if you look at like what the impact that group of people has had on modern chemistry, it's amazing. It's the who's who, Nobel Prize winners, people who've invented billion-dollar pharmaceuticals and other very, very valuable things through chemistry. Roger Adams was actually a consultant at DuPont for many, many years. And there was a very close relationship between the chemical industry and these academic big guys. There's another guy named Carl Marvel, who is again, very, very well-known chemist. These chemists were very influential in educating the next generation of professors that educated people like me. So
setting the foundation.
And Jason, it's in that continuum that, Mechoulam existed is, he came in after Wood, Spivey, and Easterfield did their isolation, after Robert Sidney Kahn figured out the structure, after Roger Adams. And if you look at Adams's papers from the 40s, you'll see structures there. I mean, he's got delta 8 and delta 7 drawn because they didn't have NMR spectroscopy to elucidate where the double bond was in the molecule. So they were just sort of taking some guesses based on the degradation chemistry.
Exactly, just how it was behaving. Yeah.
Right, how it behaved chemically. But recognize that as soon as you treat delta 9 with something like nitric acid or any strong acid, sulfuric acid, it's going to go to delta 8. So you're gonna get fooled in thinking that Delta 8 was what you started with, but actually
Right,
because
right.
Delta 9 is metastable. So that confounded researchers. But the other thing that I found was really interesting, and you hear a lot about this these days because with the hemp bill, or I'm sorry, the
Yeah,
farm bill
yeah, the
making
farm
hemp
bill.
legal, there's
Hmm.
a lot of this CBD that's being converted into THC as though it were a brand new thing. But
Yeah.
this has been in the chemical literature since 1940. I mean,
Yeah.
it was first published in 1940, and then Mechoulam followed up later in the 60s and the 70s. But it was really important because what Raffi recognized when he came in. So Raffi was, and he will humbly, well, if he were still here, he would humbly admit
Yeah.
that he stands on the shoulders of Todd
Oh
and
yeah,
of
he writes
Adam
about it repeatedly.
and of R.S. Well,
Hmm.
and the way I know that is I've probably read about 100 of Raffi's papers. I think
Yeah,
he's got
yeah,
like over 300 papers. I think I've read about 100 of them. And I can tell you, when you read the literature that a scientist writes, when he's thinking about this and constantly turning it over in his head,
hmm,
you really start to learn about the person.
yeah,
Like
yeah,
I never met Raffi. We had several exchanges back and forth, and I always just lit up when I saw that I had an email from Raphael Mechoulam. Oh
yes,
my
yeah,
God.
yeah,
Stop all the presses. Let me read Raffi's email. So it was great that he had a respect for who I was, again, based on the fact that I'm a chemist. I could speak to him in a language of chemistry, which is a language of
yeah,
structure, a language of literature references, a language of really understanding all the work that's been done before you.
yeah,
And all the work that's been done before me, well, that's the same work that's been done before Mechoulam, same work that's been done before everybody. It's this continuum of science. And the beautiful thing, I think, is that if you read Raffi's papers, he references Wood, Spivey, and Easterfield. He
yeah,
references Roger Adams, and he references Robert Sidney Kahn and Alexander Todd because he was standing on their shoulders.
yeah,
And Raphael was a good enough physical organic chemist to understand that, you know what, we're isomerizing this double bond. So let me isolate the active
hmm,
ingredient in the most gentle way possible to not isomerize the double bond. And now we can use this new tool, which was
let's
nuclear magnetic resonance, which was a way of looking at the different magnetic signatures that different hydrogens have in organic molecules. And you could run an NMR spectrum and you could tell based on the peak shape
hmm,
and based on where the peak position is, what kind of hydrogen. And so he was able to figure out that the delta 9 configuration is the correct configuration that occurs in the plant. And so Raphael's big discovery here is not necessarily the first to isolate THC.
exactly, yeah,
Wood,
right,
Spivey, and Easterfield did that. And if you look at Wood, Spivey, and Easterfield's boiling point data,
yeah.
which is just as good today as it was in 1896, it is spot on
Yeah.
Raffi's numbers. I've
And there's
showed you that
and there
the
was a
numbers,
paper that actually
right?
came out, I think a couple years ago that also specifically addressed the boiling point issue and looked at the old literature, compared the, um, like, is this reporting at atmospheric pressure or under vacuum?
Right.
And I can't remember the name of it, but I'll try to remember to throw it in the show notes because it came out after you and I did an episode talking about some of this stuff. And so
Yeah,
now there is a review publication that tries to
that's right.
synthesize all of that. Thank goodness,
And
because it
and
is a
and what's
mess.
the boiling point of THC? Spot on exactly what I had said in my article, which is it
Yeah,
should be about 427 degrees C. And that's exactly what they came up with. And the beautiful thing is, so this is why the literature is so great. When you look in the literature, people are they're trying to write about the science they're doing to the best of their understanding.
yes, yeah,
But as that frontier moves and we develop more and more understanding, now you can go back and look at their data and say, you know what? That wasn't cannabinol they isolated. That indeed was THC that they isolated. So then the first thing that, well, they'll say, well, Raphael discovered THC. Well, he didn't discover THC. In fact, he didn't even discover the structure of the formula. That we contribute to Robert Sidney Kahn. Yep. The first to isolate was Wood, Spivey, and Easterfield. But what Mechoulam did that was very, very important, and this can't be understated, was he used the new tool of nuclear magnetic resonance, NMR, to correctly elucidate. Elucidate is the $1 million word here. Yep. To correctly elucidate the structure of the active ingredient taken from the plant. So if you read his 1964 paper, he takes hashish, which I think he got from Lebanon.
Smuggled,
Lebanon. He talks about it in his movie. It's really funny. And then he does he column chromatographs it. Well, we know that if it's hash, it's most likely THCA, right? Not THC. And then so he chromatographs it. So he separates out the minor cannabinoids and other things and just takes a fraction of what he believes is the major active ingredient. And then he took that material, distilled it, and then made a crystalline derivative, as often is the case. If you could make a crystalline derivative and then you could purify by recrystallization, that's like making rock candy, right? You just wash away all the impurities and the only thing that's left is your crystalline pure substance. Well, then what he did is he removed the auxiliary group that made that crystalline. And now we know that this is pure Delta-9 THC for the first time. And when he ran the NMR, he was able to show that the vinylogous proton is the number 10 one, because he can do coupling. Coupling is how a certain proton will interact with adjacent protons, and how that couples gives the chemist the idea of what the structure is. And so using NMR, he was able to correctly elucidate the structure. That's the contribution in 1964. It wasn't the first to identify, it wasn't the first to isolate, but it was the first to elucidate the structure, and that's extremely important.
And it started with, so a couple of things important to highlight here. One, so, the research you were talking about with Roger Adams and everyone, that sort of stopped around the 40s when we got into World War II and Prohibition and all of these things. And then in the 1950s, you had NMR that was developed, but then you also had gas chromatography that really, as we know it today, really came online. And it's worth noting that too, because gas chromatography gave researchers a way to very quickly separate and get some idea of the constituents of a compound much faster than, it's good to put this in perspective for people, that the columns that chemists were using back in the early 1900s were like these huge custom-made columns that, you can find pictures of them that are really awesome, where you can see people standing at one end, pouring the mixture in, and someone at the bottom with collection flasks changing them out according to, time and everything. So this technology, it really did evolve quite a lot very fast. And so the 1960s, I think the first commercially available GC was like 1959 or something. It was pretty late. So really the 1960s were the the first opportunity for these tools to be applied to these questions. And I think that's, Rafi does a great job in his writing of pointing out that all he was doing was getting back to the problem that all of these other researchers were trying to answer, which is, how does cannabis work? We know that it works because, going back to William O'Shaughnessy, we know it has these effects. We know, there's interesting potential and possibilities, but how does it work? Why? What's driving all of that? And so, like you mentioned, it's the application of these tools, NMR as well as GC, because that just allowed people to do things very fast compared to what they were able to do before in investigating complex mixtures. And it wasn't just cannabis. This blew natural products research wide open.
That's right.
Across the board. But it's great to use cannabis as the example of really what was a trend. I mean, the '60s was a very exciting time for chemistry, to have these new tools, and people that like Rafi that were in school in their post grad programs or whatever, and starting to get access to these things. I mean, it's amazing to read because you see just a huge explosion of publications
Yes.
around these things, identifying, elucidating these structures for all sorts of things.
Yeah.
So not just in the cannabis story, but also in the broader natural products chemistry story,
Yeah.
Rafi plays a big role in that too.
Yeah, absolutely. I mean, most notably, gas chromatography is used today extensively to look at the essential oil aspect, or the terpenes that are in cannabis extracts. And so the first report of terpenes isolated from cannabis was again by Alexander Todd back in the 30s. Terpene chemistry is just a whole new thing. It's just like, actually, it's a whole old thing.
Yes, a rediscovered thing. Yeah.
There's a book that was translated from German in 1902, I do believe. In fact, I'm trying to remember, Otto Wallach, I think, won the Nobel Prize in chemistry, I believe, for his discover of alicyclic compounds, which is what they called them back then, but the terpenes. And if you look at his treatise, which I think was in German, but then was translated into the English, that book, which was written in like 1902, is still relevant today. Terpene chemistry really hasn't changed too much. And like you said, Jason, with the advent of gas chromatography in the early 60s, the first report in the literature, in the chemical literature, of the analysis of the terpenoids and terpenes found in cannabis was done in 1961 by a Canadian group who published in Nature. And that's the first time you see a chromatogram of the terpene profile of cannabis essential oil.
They wasted no time, because that's one of the earliest times you could even do it.
Knew right away.
Yeah.
You can use this tool to figure out what's in cannabis. And today, terpene profiles are measured by GC, either direct injection or headspace measurement using gas chromatography. So, but you're absolutely right. What the advent was in chromatography in general was, so the way that gas chromatography separates compounds is based on their different boiling points. So each pure compound have unique boiling points, and the way a gas chromatography works is it's basically separating things so the most volatile ones come out and are detected first, and then the less volatile ones come out later. And so in this fractionation of the terpenes, you could do the same thing with cannabinoids, but recognize that now you're going to be looking at decarboxylated cannabinoids, because the injection port temperature that heats up the sample that goes into the gas chromatography column is hot enough that you'll get decarboxylation. And again, maybe there's something to that, because the cannabinoid acids weren't discovered until some of Mechoulam's later work. Mm-hmm. So remember that when you distill a cannabinoid acid, it irreversibly decarboxylates. So what you're going to get as your product in your distillate is going to be decarboxylated cannabis. But at some point, Mechoulam said, hey, you know what? There's an acid group here, and figured out that THCA in the trichome is not THCA, but it's THC. I'm sorry. THC is not THC. It's THCA. It's made as its acidic form. So that was also Mechoulam's work. And so I think Raphael recognized at the get-go that there was some ambiguity about the position of the double bond. And that was something that was first observed by Robert Sidney Kahn, later by Adams and Todd. They all knew that this double bond walks around. We don't really know where it's at in the plant. But Mechoulam, that was his first and most important, I think, contribution early on.
And it
After
really
that, what he
set
did
things off
was,
to the races from there.
well, what he did was he really wanted to investigate that delta 8, delta 9 thing, like maybe, and in fact, it was Mechoulam who followed up on the initial isomerization work that Roger Adams published 20 years earlier in the 40s, where Mechoulam now understood the roles of the acid, that the acid plays in making THC from the cyclization of CBD. Yeah. So in the case where you're using a protic acid, so a protic acid is an acid that actually has a proton. It has an H plus. And so you can think of like sulfuric acid is a protic acid. Acetic acid is a protic acid, right? Because the proton is actually the acid. Yeah. But what Mechoulam found is that Lewis acids actually work. And now a Lewis acid is not a protic acid. It's not a chemical species with a proton. But what a Lewis acid is a Lewis acid is an electron pair acceptor. And so this is a very important and very fundamental difference, is that when you isomerize CBD with a Lewis acid, and Mechoulam has used BF3 quite a bit, boron trifluoride, right? That's B, boron in the middle with 3 fluorines. Well, those 3 fluorines are highly electronegative. In fact, fluorine is the most electronegative element on the periodic table. So what you're doing is the fluorines withdraw electron density from the boron in the center. And boron only has 3 things around it, right? Which is weird, because in chemistry, you strive for something called an octet, which is 4 things around it. So what that means is that boron has got an empty p orbital. Cool, right? The empty p orbital is basically an electron acceptor, because the fluorines are inductively pulling electron density away from the boron atom, which basically brings electron density into that empty p orbital. So when you take BF3 and you treat it with CBD, the first thing that happens, Jason, is that the oxygen, one of the phenolic oxygens, attacks the boron, or the unshared pair electron now coordinates to the boron, and that makes a trifluoroboroalkoxonium ion. But anyway, now the acid is the proton, the phenolic proton. So what happens is when you coordinate that phenolic oxygen to the boron, that proton that was on the oxygen is now super acidic. Yeah. So in a way, what the Lewis acid is doing is it's creating an internal protic acid.
By
reacting with the phenolic groups, right? Did I explain that okay?
I mean, I got it. I don't know for folks that don't that haven't studied chemistry in a while. Some of that might be a little over their heads, but I definitely understand. I understand what you're saying, that it's almost like a prodrug in a sort. That it's through the interactions that happen, you're freeing something else up that then can act as that that acid. If that's kind of a more simplified way to,
you need a proton to undergo the cyclization.
Right.
So you'd say, well, where's the proton with BF3? In the case with BF3, the proton is already on the CBD molecule, right?
Yeah.
It's one of the OH groups. And so when that OH group, when the oxygen from the OH group reacts with the boron, it just forms a complex, right? It doesn't actually form a bond. It's a non-bonding
Right. They just want to be close to each other.
Yeah. I used to have a chemistry professor who said, the bonding interactions, we know those. It's the non-bonding interactions that makes chemistry exciting, right? Because
Yes,
when you
yeah.
get non-bonding interactions, they're kind of like off and on, off and on. So
Right,
you can
right.
imagine that
Yes.
the boron kind of sits on that oxygen and now makes that proton super acidic. So now it becomes the acid.
Yeah.
That protonates the double bond, and that gives a carbonium ion, which then closes with the oxygen to make the third ring of THC.
Yeah.
So Mechoulam's contributions there were so important because what he found is that not only do you make THC that way, but you make this abnormal isomer called iso-THC, right?
Yes.
So the
Yeah.
first
Yeah.
one to
Yeah.
describe that was Mechoulam. And then later on, a researcher named Raj Razdan, who was at the Arthur Little Company, who they were doing all kinds of interesting cannabinoid stuff. He studied that reaction to the hilt. So they were thinking, I think, along the lines of making THC versus extracting it from cannabis,
Yeah.
and how can we make it through this cyclization reaction. And so what you find is that when you use a boron trifluoride acid, and this was published by Mechoulam and followed up by Razdan, is that you get a statistical mixture of THC and iso-THC.
Yeah.
Iso-THC being the abnormal isomer.
Yeah.
And here's something really cool, Jason. This is totally new. Like, this is not in the literature, and this is new science. So Cayman Chemical sells an analytical standard for iso delta 8 THC. And iso delta 8 THC is one of the major non-natural isomeric byproducts that's in delta 8 that's being produced today. And what complicates this in delta 8 production is that the iso delta 8 coalutes with delta 8.
Yeah, it all looks the same.
It coalutes by HPLC, but the folks at Cayman Chemical just last week published a method to separate these things by GC. So there's an opportunity for GC that HPLC is unable to separate these isomers from one another, but we could separate them by GC, which is fantastic. And hopefully that will lead to a development of a standardized method to use gas chromatography for the quantification of delta 8 and the delta 8 isomers that are formed in the acid catalyzed reaction with CBD. That was pioneered by Dr. Richard Sams at KCA Labs, and I think he coached Cayman on that method. But I expect that there will soon be a standardized method for all delta 8 analysis where it's being run by GC, which is totally cool. But here's another little piece. So when Cayman announced that they were selling this analytical standard of delta 8, I got a good relationship with the guys at Cayman, and I was able to basically shake a free sample of a whole 5 milligrams.
Oh, wow. Nice. Yeah.
Away. But, what am I going to do with iso delta 8? So what I did is I just had them send that sample to Alan Howlett. And I explained, I explained to Professor Howlett. So for your listeners who don't know,
another big part of the story coming up.
Yes, yeah. Alan Howlett is the molecular pharmacologist or I think that's what she is, or neurobiologist or whatever.
Certainly, neuropharmacology is a lot of what she's done.
That's what it is.
Yeah,
who discovered the CB1 receptor. And looks at different compounds and how they act on upregulation of phosphorylation and other signals that are involved in cannabinoid binding. And so we have a paper that a postdoc is giving at the ICRS later in June in Toronto on the results of our study. And what I can tell you is that it's active.
Nice,
this is brand new science. This is really cool. What we're showing is that the iso THC isomers do show some of the hallmark signs that cannabinoids do in enhancing cellular signaling.
Yeah.
So stay tuned for that. When I could talk more about that, I will. But that's really exciting to find. But going back to Mechoulam again, I think Raphael, certainly being the chemist that he is, looked at all the cannabinoids and looked at, I think, everything from CBC to CBN to derivatives of CBD. I know in his later work, he was talking about the methyl ester of CBDA being a more stable compound than CBDA. CBDA is highly prone to decarboxylation, but the methyl ester is not. So he's able to show that the methyl ester can be delivered into the body, and then
the body
in the
pops
body,
that off.
It undergoes hydrolysis like the prodrug you mentioned, and it is able to act as CBDA. So I think that was really important on his chemistry thing. But his most important work to date, I do believe, came out of his fascination on how do these things work. And we talked about Professor Howlett. So being early on in the game, I think they assumed that there was maybe a non-specific target. They didn't think that there was a single receptor.
Right. They thought it might be enzymes even potentially that were targeted.
It's acting prophylactically or peripherally. It's just not. It's not.
There was talk about maybe it affects the fluidity of cellular membranes, like
or something like that. Right.
Yeah.
Right. There was all kinds of theories put forward, but when they actually, for the first time, identified the CB1 receptor, it really opened up the whole field of endocannabinoids.
Exactly. Why are those receptors there? Yeah.
Why are those receptors there? Right. Those receptors aren't there for an exogenous compound that comes from a plant.
Well, let's actually highlight that, because Raphael Mechoulam actually says that multiple times, and I think it's worth reiterating because this is a legitimate question that a lot of people ask me. Do we have cannabinoid receptors because of cannabis? And so I want to take that question seriously, just because a lot of folks, especially not even just folks that are newer to cannabis, but I get, like fairly established people in the industry that ask me that too. And yeah, the assumption is not immediately that we have receptors because of cannabis. The assumption is that there are some endogenous compounds within us that we don't understand that the body's use that for. And Rafi I think I even have one of the papers up here. I'm trying to remember exactly what he says, but he says something like very casually, like describing the history of discovering cannabinoid receptors and endocannabinoids. And he says, of course, these are not here for exogenous compounds. There's more to the story, and we just have to dig a little deeper because clearly we don't understand our own human physiology as well as we thought we did.
Well, that goes without saying. I mean, I think in Rafi's movie, he talks about it at the very end that the endocannabinoid system is probably one of many molecular-based receptor systems that are in our bodies. I think the dopamine or dopaminergic system that's in the brain was well understood. Well, not well understood, but it was explored and looked at. And I think there's an interesting interaction too between the dopaminergic and the endocannabinoid system, because dopamine is that reward. That's when you feel good and when things are going. And certainly cannabis can get in that realm too as well. And then you got the 5-HTAs. I mean, that's just like there's this whole library of receptors in your brain.
All the serotonin subtypes, and
it truly is a combinatorial problem because you can't affect one and not affect the other because
it's complex systems. Yeah, they're all connected.
Right.
Yeah,
right. They're so woven together that it's really difficult to just discern what any one single thing is doing.
Well, and
let
it's
alone
it's worth
all of them.
highlighting that too, because I think some people tend to think that, um, when they're trying to engage with cannabinoids or cannabis, that it's more or less a kind of simple thing that they're essentially feed the term often used is they're feeding their endocannabinoid system, which can be true to an extent. But it's also important to note that influencing the endocannabinoid system inevitably influences all of these other systems too.
That's right.
And so you have to have some respect for just how interconnected that all is. And you're not just interacting with cannabinoid
No,
receptors and TRPV1s and stuff in isolation. There's all this other stuff going on. And so it's
that's right.
important for folks not to, it's so easy for our brains to oversimplify pharmacology and a lot of these concepts, especially when marketing makes it very hard to discern what's real and what's not, because companies that produce these products will make it sound very simple and straightforward. As just another point I want to highlight that,
Well, there's a transient nature there,
yeah,
and things are constantly changing. And a theory that I have, which is borne out in some research but still needs to be further researched, is that one of the variables of the entourage effect is that cannabis in its natural form
mm-hmm.
is much more effective than the isolated cannabinoids themselves. And so I think that it's highly probable that the wild card in the entourage effect is the rate and magnitude of upregulation of receptor expression on the cell surface.
Yeah,
And so it's known that there are signals that come into the cell that work on the nucleus that can upregulate enzymatic production or can
right.
upregulate the synthesis of receptors. And I think, it's very difficult to kind of flesh this out because there's so many different things going on.
Mm-hmm.
And like you said, the
It's hard to measure.
interconnectivity of these systems. But what's known in general is that terpenes and terpenoids that are found in cannabis extracts modulate the activity of the cannabinoids. So you could consume a cannabis extract that maybe has a lower amount of THC but has a lot of terpenes, and you'll feel a much larger effect. I'm sorry about that.
Oh, that's
Than
okay.
if you were just consuming the cannabinoid itself. I think one of the things that bears that out is the fact that when you look at plant medicine, where there's multiple components, right? There could be the terpenes, there could be the active ingredients, in this
Mm-hmm.
case, the cannabinoids. There could be lipids, there could be sugars,
Right.
there
Yep.
could be what they call the full spectrum extract, right? Which is
Yeah.
as much as you could bring from the plant. And I think, anecdotal evidence says that, well, those types of preparations are more active than purified cannabinoids. Well, what's the molecular basis of that? Right?
Yeah.
What's the biochemical basis? And so the theory that I have is that terpenes are known to increase fluidity at the lipid bilayer, right? The lipid
Yes.
bilayer
Yes.
are the
Yeah.
cell walls
Mm-hmm.
that protect the inside of the cell, the cytosol portion of the cell, from the outside. And so what happens is terpenes kind of, I don't know if the right word is intercalate, but they can get in to the lipid bilayers and start opening up channels, which starts exchanging compounds on the outside come in, start, compounds on the inside go out. I liken it to opening the windows at your house. When
Yeah.
you
Yeah.
open the windows at your house, you're going to start exchanging the outside air with the inside air. So what I think, Jason, and again, look at some of the Mechoulam's data, and you could look at some of the clinical data with cannabinoids where they're looking at whole plant extract versus single isolate. And what I believe is that the entourage effect is potentially somehow enhancing the uh, uh, formation of the receptors themselves, right?
Yeah.
Because you can imagine that if you have a certain amount of THC, well, if you have a lot more receptors, you're going to get a lot more activity
Yeah.
because
Yeah.
there's lots of baseball gloves to catch that ball, right?
Yep.
But certainly, like if you look at like where you're getting, I don't know, like I know a lot of people like cannabinoid isolates, like CBD is very popular in
Sure. Yeah.
certain beverages. And certainly, like if you extract CBD isolate, which is a white crystalline compound, it doesn't have all of the earthiness that comes with the whole plant extracts. And certainly, like if you're drinking like a soda or having a beverage or
Right.
something,
Right.
you
Yes.
may not
Yeah.
want to taste all that
It's acrid.
earthiness,
Yeah.
but you still want the effect of the cannabinoid. So I think in today's marketplace, there are single active ingredient products that I guess people seem to enjoy. But plant-based medicine should really have the whole plant in there. And I'm not saying that you have to have full spectrum whenever you take cannabinoids, but I think if you're trying to get the medicinal benefit out of it, there's still things that we don't quite understand
Right. Yes.
how
Yeah.
these things work. But it's like, well, there was a really fascinating paper. It was out about 5 years now that looked at the plasma blood levels of CBD, and they looked at an extract that had all the minor cannabinoids, terpenes,
Right. Right.
maybe fats and waxes, and they looked at CBD isolate. There was no comparison.
Yeah.
There was
Yep.
almost a 4x difference in plasma blood levels taken from a whole plant extracts versus isolate. So I think Mechoulam's notion that he kind of coined with Ethan Russo around the entourage effect goes way beyond cannabis, right?
Oh
It
yeah.
goes
Yeah.
herbal medicine in general. I mean, we've gotten analgesics out of natural products. I mean, the most typical would be aspirin, right?
Yeah.
Which is
Right.
acetylsalicylic acid. Well, salicylates are in willow bark, so
Yep.
maybe willow bark extracts might have better analgesic properties than just the aspirin itself. But I don't see anybody chewing on willow bark or
Right. And
anything
it all
like that.
depends on the pharmacology of some of those accompanying compounds. Like cannabis is nice because um, the herbal synergy works in a sort of more or less positive way. Although there are some examples of medicinal plants where, to effectively engage them, you do want to prepare them in some certain way in order to alter the chemistry a bit to get, potential toxic elements out or whatever. But I definitely agree that a lot of that, there's a lot that we don't understand around how this herbal synergy works. And something I've wondered, um, so a couple of things. One, the cannabinoid acids influence absorption and bioavailability of the decarboxylated cannabinoids. And that's something that I think plays into these synergistic effects. And then what you were saying about terpenes playing a role in affecting how things can move across membranes. I've wondered about, there are these transport proteins that, depending on certain conditions, they will carry things into the cell, and they will carry even cannabinoids, and things like that across these cellular membranes. And I've wondered how different types of terpenes, particularly looking at like your alcohol terpenoids, with those hydroxyl groups versus pure terpenes, how they drive those effects. Because I would imagine if that's what's happening, then you should see some correlations there between those sort of classes, because they're going to have different, because of the hydroxyl group, they're going to have different behaviors in terms of their ability to
Yeah, the
affect those
hydroxyl group
membranes.
the hydroxyl group can conjugate to a sugar. You can functionalize the hydroxyl group where a this is often a sticking point that I often have with people. You know, the wider class of compounds are terpenoids, right? Like cannabinoids.
Mm-hmm.
Terpenes are a class within terpenoids that, you know, strictly speaking, are oligomers of isoprene, and they're just hydrocarbons. So as soon as you introduce an oxygen or a sulfur or some other heterofunctionality, you no longer have a terpene, but you have a terpenoid.
Right.
And in the case of cannabis, it's so interesting because when you look at freshly harvested cannabis, the most predominant terpene in many, many strains is myrcene.
Right.
And myrcene is kind of like, I don't know, I won't call it the CBGA, but it's kind of like the stem cell terpene. Because
it is, yeah.
Yeah. Okay. So you know
I know what
I'm
you're talking
saying.
about because I've looked at the biosynthetic route so much. I know where you're going.
Yeah. So basically, so if you look at the biosynthetic route, you're looking at GPP, right?
Yeah.
And so if GPP just loses phosphoric acid and makes a double bond, so what happens is the N carbon's got the phosphate group, and then the beta carbon's got a proton. So what happens is it loses phosphoric acid, generates a double bond. That's myrcene.
Yeah.
That can also, so if you lose the phosphate group, you generate what's called a carbonium ion. Well, because there's other double bonds, those double bonds can rearrange in that carbonium ion to give you the pinene skeleton, to give you the thujocene skeleton, to give you the limonene. So all of these terpenoids basically come from common precursors, which is really, really cool.
Yeah.
There's very, very notable work by Professor Paul Malberg at the University of Indiana, who did the lion's share of what we know about cannabinoid biosynthesis in the trichome. And when you talk about accumulation of cannabinoids and terpenes in the trichome, you should look at the work of Paul Malberg. He just did some fantastic stuff where they looked at, you know, where the cannabinoids are being formed. They looked at different, you know, are there cannabinoids in the stalks? Are they just in the sessile trichomes? They did some incredible microscopy and staining work to basically identify where these cannabinoids come. But going back to Mechoulam for a second, how this impacts him. So I think Rafi was a classically trained organic chemist. And at the time, you know, again, still to this day, you know, the FDA approval process for approving drugs is based on a single active ingredient in basically a matrix of inactive ingredient. I mean, that's the monomolecular medicine that FDA. So when you talk about cannabis and you say, well, there's more than one active ingredient, and then there's this synergy between the other non-cannabinoid components, it basically turns that whole monomolecular medicine thing on its head, because as scientists, we want to study a single active ingredient and how that interacts with the body. And when you look at these FDA approved drugs that, you know, if you ever watch the evening news at night, you'll find out that
the
brought to you by the pharmaceutical companies and their commercials of the litany of side effects, while they show some family frolicking,
smiling, and bright sunny day. Yeah.
Right. I never knew what the skin of the perineum was until I heard it on a Farxiga commercial, I think, or the crazy names they come up with these drugs. But a very important contribution that Raphael made was they recognized that CBD can be used to control epilepsy. And so they did a trial in Sao Paulo, Brazil in the late 70s and early 80s, and found a high therapeutic efficacy for CBD in treating difficult to treat seizure conditions like Dravet syndrome and Lennox-Gastaut. And there's a couple other kind of like types of epilepsies that just don't treat well with the current anti-epileptic medication. And what they found with CBD is that it worked across the board, and it worked really great with little to no side effects. But then, the thing just kind of was out there in the literature that
Was that the
CBD.
study where they got like almost 100% response rate or something? There was one study they did. I don't
Yeah,
remember if it was that
I
one or
think
not.
the study you're referring to, which he mentions in the Scientist, is treating pediatric nausea associated with cancer
That's
treatment.
right. Yeah, you're right.
And in that case, in dosing with a very small amount, I mean, we're talking micrograms amount of THC under the tongue of infants, it completely blocked all chemotherapy associated nausea. And if you've ever had chemotherapy or know anyone who's ever been through chemotherapy, it makes you nauseous. It just makes you sick. It just makes you not well. So, this was where GW Pharmaceuticals came along, like 20 years later and said, hey,
Yeah.
looks like CBD works on epilepsy. Let's try this, right? And it's interesting to note, Jason, that rather than doing a whole plant extract, even though they grow plants to make their CBD, they're still purifying it into the isolate form, or pretty pure.
For Epidiolex.
And then just putting some flavor in there, and that's the product. So you would think that in this day and age that we would have a way, especially after all the work that's been done in cannabis, to adopt botanical medicines in a different mechanism of an FDA approved drug. And certainly like what's happened recently where they've turned down Charlotte Webb's, petition to list it as a dietary supplement.
Yeah, it's not surprising.
It makes me believe that the people in our government were really snookered on the Farm Bill. I think they were just thinking seeds and rope and paper. I don't even think they even contemplated CBD, let alone contemplated psychoactives from hemp that can be made from CBD. All that literature was known. I'm sure there was no contemplation that, we're going to let people grow hemp and everything's going to be fine. And it's like, with the price of THC the way it is today, Jason, and the price of and the availability of CBD, it is cheaper to convert CBD into THC than it is to extract THC from cannabis plants. And we're in a commodity market where price wins every time.
Yep.
So if you have a more economical way to make your product, that's the way that's going to.
And apparently you don't have any barriers, because the whole thing with the hemp system is that these products are being made. I mean, straight up Delta 9 THC is being made and sold under the auspices of the Farm Bill. It's hard to imagine why people wouldn't do that. It's like, oh, you mean I don't have to jump through the regulatory hoops of these legal cannabis markets and have to have, meet all these extra security requirements and traceability, all of that sort of stuff. And, I can get my products into gas stations and grocery stores and, wherever else.
Internet without asking age.
Right. Yeah. I mean,
age.
It's something that it makes a lot of sense for people that all they care about is making as much money as possible on THC. It makes perfect sense why they would do that. And something that and I know we're getting off track a little bit, but it's just fascinating to me. And just from a chemistry standpoint and sort of the cultural side of everything. I was in Florida recently trying to help legislators understand the consequences of some of the language of some bills that they're putting together to try to regulate Delta 8 THC products.
Yeah, they're trying to regulate it down there, right?
And they were teaching me about just how big some of these Delta 8 companies are. These hemp-derived THC companies are, just how huge they are. And a lot of them moved to Florida over the last couple of years, because they view that as a place they could operate safely in terms of being able to um, do business as they want with minimal regulation, interference and everything. And I mean, I knew it's one of those things of like, but then once you see it and you really hear the stories and understand, I mean, this is huge, huge. Like these, the THC market in the hemp side has become dominated by some companies, some groups that have deep, deep, deep pockets, and they are really fighting to, keep this going. And it's just way bigger than I imagined it was, and I already imagined it being pretty big. But there's just a ton of money involved in ensuring this all continues as it
is
has.
it any wonder that those markets are flourishing in states that don't have Delta 9, right?
Exactly. Yeah, yeah.
So
Like here in
it's
Mississippi, you see these CBD stores everywhere. And why do people go to the CBD stores? It's not to buy CBD. It's to buy Delta 8 products. And that's what have a lot of folks ever since I've moved back to Mississippi. I've had a lot of people show me C of A's and product labels and stuff of these Delta 8 products they're buying. And all of it is suspect. Like most of the COAs look fraudulent to me, or they are associated with batches from years ago that have nothing to do with anything that's being produced. And I just keep reminding them, I'm like, the only reason this exists is because cannabis is, well,
prohibited.
medical's legal now here, but it's still prohibited. And it's like, if cannabis wasn't prohibited, none of this, there'd be no need
That's
for
right.
any of this.
That's
Um,
right. And we wouldn't know what Delta 8 is. We'd think it's a version of COVID or something. Or yeah, I
right.
mean, there's
Yeah.
no
Yeah.
there's no mistake about it that people want THC. They want to get high. They want to feel the euphoria. And when when you could make THC from CBD and then dilute it in a
Yep.
gummy back down to that 0.3% or
Which in a
lower,
gummy is actually quite a bit, milligram per gram wise.
yeah, I mean, you could pack, upwards to 10 or 15 milligrams, depending on how big your gummy is,
Exactly. Yeah.
and still be below that threshold. But the real disappointing thing to me is that yeah, there is big money, and one of the reasons why there's big money there, Jason, is it's a much larger addressable market,
Right. Yeah.
right? And so you're stifled in a single state when you have a license in that state to only sell in that state,
Mm-hmm.
where these guys can sell everywhere
Yeah.
across the United States and on the internet. And you'd think too that, well, like I know that I think Mississippi didn't do this, but I think Georgia and Alabama are doing this. They want to regulate a cannabis, but they want to regulate some low THC bullshit version
Mm-hmm.
of cannabis. And like with Alabama, I think you can't be above 10%. I think in Georgia, you can't be above 5%. And
Yeah.
it's like,
Mississippi chose 30, so they did all right.
well, here in Vermont, and we have adult use here in Vermont, they've capped the THC potency at 60%. I don't
Oh,
know
and
where they get
extracts.
the
Yeah,
60%
Mississippi did
number,
that too. They chose 60% for the extracts. Yeah,
but it's
30% for flour.
ridiculous because you're lucky if you can get a rosin to test at 60. That means you have to basically do fresh frozen to get a huge terpene fraction to dumb down your
Oh, exactly.
THC.
Yeah. Or they're blending in
So
CBD to push it down.
yeah, that sucks too,
It
right? To
does
kind
suck.
of
It sucks. I hate that it happens, but yeah. It's
right. So I went to the dispensary the other day and I noticed that there were two rosins and I bought them both, right? Full melt rosins. They were right at that 60% mark, right?
hmm
And so does it taste as good as like a good THCA BHO? Not even close.
yeah.
In fact, like you say, I think they actually have to add stuff to dumb things down, which is just the opposite of what you want to do, you know?
Yeah, I know.
But
It's what I've been trying to encourage people here is like to think about that broad chemistry, and it's like rather than trying to add stuff back in, see if there's a way that you can try to um, have your extraction be as broad as possible without getting into like chlorophyll and all of that. But like you were saying, like get the widest terpene fraction you can, get the widest series of minor cannabinoids and everything that you can to reduce that need. But
yeah,
it's
I
just
think
tough.
closed loop hydrocarbon, in my estimation, gives the cleanest extract.
It's just a little harder to scale.
I've seen some rosin profiles that look pretty good, but in general, the problem with rosin is that the scalability and the loss is a production nightmare. It just becomes very difficult to scale. But with closed loop
Hmm.
hydrocarbon, that scales beautifully. And I could show you COAs where we could account for almost 100% of the extract by just adding up the terpenes
Yeah,
and then adding up the cannabinoids. We get damn near close to 97, 98% of the stuff accounted for. That means there's only like 1 or 2% of stuff that we don't know what it is. That's pretty good for a plant
yeah,
extract.
yeah. Absolutely, absolutely.
And they taste great and they're extremely effective. So, if it ain't broke, don't fix it, man. That's the
I
way to
know, yeah,
go.
yeah. I've never been a fan of potency limits. It is interesting on the flower side. What's been interesting to see here is there's no rush to try to get THC numbers above 30 because you can't because it's not allowed. So there's not the same sort of pressure on labs to boost numbers, at least to absurd degrees. There's pressure on them to boost them as close to 30 as possible. But
Yeah, that's all. There's no cannabis flower
no,
on planet Earth that has 30% THC. If you understand the structure of the plant and where cannabinoids are coming from,
yeah, no,
in fact, I even think 20 or 25% is a stretch too.
I think that's
Look,
the
the
upper
thing
limit in my from my experience, you know.
mhm.
Yeah.
The thing that's going on with testing, which is a real boondoggle, is the fact that because we're in a prohibition market, um, potency is a way of grading the value of cannabis, right? And so,
Yeah.
like, if your potency is higher, you're going to get a higher,
Yeah.
number of dollars for that pound versus something that's got a lower potency. The issue really is, Jason, here, my point of view is that reporting potencies on a dry weight basis is the issue. And that is because cannabis is not dry.
Right.
Cannabis is moist, right? Even if the stems crack, there's still moisture in there. And so the difference in moisture content, if you think about it, if you're reporting on a dry weight basis, that means you need to know in your denominator,
Hmm.
what does that bud weigh without zero water, bone
Right.
dry, no water? Well, check it out. These labs can't dry that herb over a period of 2 weeks in a desiccator. They dry it overnight
Hmm.
in an oven by heating it up.
Yeah.
Well, guess what happens to weed when you heat it up? The terpenes start
Yeah.
volatilizing, right?
Yep.
You start decarboxylating cannabinoids. So what you're testing on that bone dried
Right. It's
sample,
not. Yeah.
you've lost a lot of your mass. Guess what happens to the THC number?
Yeah. It's gonna
It
go
goes
up.
up
I
because THC.
actually did a study on this that I cannot release publicly because it was for a private company and that's just how it goes. But I actually did a research study on this that I wish I could release the data from, but I'll explain it. I don't think there's anything wrong with me explaining it. Um, I did a really long, gentle loss on drying study to look at this, and what for people that don't know, loss on drying is just what it sounds like. You take some material, you put it in an oven at a set temperature, and every so often you measure the weight of that material. You see the weight going down and down and down, and the idea is that you're if you're dealing with something that doesn't have a lot of essential oils or something, the idea is that you want to see that moisture content, you want to see that weight plateau. So that's not
That's right.
changing anymore. And then theoretically, at that point it's dry. However, with cannabis, if you do this, your chart will look like a staircase. You'll see it go and it'll plateau for a while. And then you'll see it go further and plateau for a while. And then you can see it go further and plateau for a while. And what you're seeing is water loss, terpene loss, CO2 loss, all of these things that you just described coming out.
Yeah.
So I'm really glad you brought that up because it's something that, one, most labs don't want to talk about and don't want to tell you. But, I'm thinking about it. I was like, I don't even think the company I work for, I don't even think they operate labs anymore. So maybe none of my worries about sharing this matters anymore. But, this is a real issue that I think labs should be more honest about. I do think it is an issue that this is one way that labs boost their numbers is by manipulating their loss on drying method to specifically exploit this issue. So it's good for people to be aware of these. I have a whole lecture where I spent an hour and a half like talking about all these different things that labs,
I'm
should be
thinking,
honest with people about.
I'm thinking that they should forget about reporting it on dry weight basis. Who needs it on dry weight? You're not smoking on dried weight. So if you're smoking as is, it should be tested as is, whatever that moisture content is. So the issue with moisture is that moisture accounts for some of the weight of that bud.
Yeah.
Right. Yeah. And so if you dry out only the water and you're not drying out the terpenes, well, we know that can't happen. Right?
Yeah, that's quite a feat.
So if that can't happen, the dried form. Mm-hmm. So maybe I don't know. Can you quantify?
It's hard.
On the as-is material and then create a correction factor
you can
for the loss of terpenes?
Yeah, you can, but it's tricky. It's like no matter how you do it, there's a trade-off because when you look at
because how do you know how much of your terpenes you're losing?
Right. Yeah.
Like if you're losing your myrcene and your osmine, the most volatile ones, but you ain't losing caryophyllene.
Right.
You ain't losing some of your more heaviers. So
yep.
At some point, so I think, just again, the way I think is that when I buy something at the store, I want to know what the cannabinoid content of that is, not that nug put into
that's a hard problem to overcome. I was actually in the last class I was teaching, I was talking about this, that we don't have test results on what you actually consume. We have test results on the material that was sampled either just after production, if it's a formulated product, or for flower, it's a test that's done as early as possible, right after cure, because they want to get that to a dispensary as quickly as possible.
That's right.
And these products change over time.
Right.
And so this idea of like, how do I get data of what I'm actually consuming, not what it was months ago or whatever. This is a very important thing people should be thinking about, and is a very hard problem to solve. Just logistically, it's tricky, which is why a lot of companies are trying to develop these like home like test kits and stuff, different devices. There's the MyDX and there's the different IR, NR machines and all these other things to try to help people test at home quickly and everything. But you raise a good point. I also want that data, and it's a tricky issue to solve, and I think it's something people take for granted. Like in some ways, we've sort of taught them to focus on data without teaching them that you also have to understand the context of the data, and that context isn't always applicable to your context, and how you're using it.
Yeah, I mean, I think potency is an artifact of prohibition.
I feel like we shouldn't even.
You don't test your beers. I mean, Cureleaf, Budweiser, my local grower here, some heady microbrew.
Right.
So, I mean, Yeah, I don't like Budweiser. I've never liked Budweiser. I've never liked American lager, but I love a good heady IPA. I love a good stout. I love microbrewed beer. So to me, cannabis is along those same lines, which
Yeah,
is
yeah.
that I don't, even though some breweries will actually give you the specific gravity of the
Right,
different
right.
beers and
Yeah.
try to give you some, you you want that IPA because you like the way the IPA tastes.
Yeah.
You could care less about how much alcohol is in the IPA. If it tastes really good, you're
Yep.
going to enjoy it. To me, cannabis falls in that same thing. It's more around not potency. We know it's going to be potent. These things make a lot of THC. The real question is, how does it taste? And what is your experience?
Yeah.
And that's a very subjective thing because what
Yes.
you might
Yes.
like is not what I like. I like my wife is likes the blueberries, likes citrusy, lemony things. I like the gas. I like the
Yeah.
diesels and chem dogs. So, I mean, our flavor palette and the olfactory spectroscopy that our
Yep.
nose can do is so sophisticated that we know what we like. I just recently tried a strain that I hadn't tried before called Animal Mints.
Oh, yeah.
And I'm
Yeah.
like, Animal Mints, wow, this is like, it's kind of like, it's a little bit like Girl Scout, but it doesn't
You're right.
have that minty thing. To me, it's more gassy and I love it. And I predicted, you know what? I bet you my wife doesn't like the Animal Mints. So I was like, hey, what'd you think of the animal? Nah, that's not what it's like. Yeah.
Yep.
So you can kind of predict like what different people are going to like almost just based on their palate.
Yes.
And
Yeah.
to me, it's like once cannabis is not prohibited and once there aren't, make or break deals based on a THC number, it's going to be all about quality,
Yeah.
which is a very subjective thing,
Yes. Yeah.
I want my buds to look fresh. I want them to smell fresh. And I could really care about the THC number. I mean, as a scientist, I'm interested in cannabinoid profile,
Yeah.
but I don't think it should be as important in the commerce of cannabis.
Yeah.
But that to me is an artifact of prohibition.
Yeah, really, really, that THC number should only matter in the context of formulation. Where you're trying to actually get a dosing, that's
Right.
consistent or whatever. And for people with specific, therapeutic contexts where that dosing matters, and they need to know what the dose is.
Right. Like in a gummy, you want to know that that's a 5 milligram gummy
Right.
and not a 20 milligram gummy. I
Yeah.
mean, their testing matters, but guess what? In the gummy, you don't have to deal with the water
Yeah.
issue and the bud as received.
Yeah, don't even
But
get
I
me
think-
started on testing data around edibles and that sort of thing, because I've
Well, what I can tell you with emulsions is that when you have emulsified cannabinoids,
yeah,
in order to measure the amount of cannabinoid, you have to quantitatively de-emulsify that.
Yes.
Meaning that you have to, so those emulsions basically are dispersed particles
Right.
of fat that are basically the reason why you can't see through this vial is like with
it's a
milk.
colloid.
Well, I'm sorry?
It's like a colloid.
Yeah. It's like a colloidal suspension. That's exactly what it is. And so it's kind of like milk. You can't see through milk either, right? Because milk has glycoproteins that basically solubilize milk fat within the continuous phase of water. The interesting thing about milk is you could continuously cut it with water and it never oils out. Right? Never oils out. It tells you that the balance between the emulsified fat and the fat particles themselves is perfect,
That's
right?
cool.
Because you
Yeah,
can't
that's really cool.
denature milk by adding water to it. But the funny thing, I wouldn't say the funny thing, but like the popularity of gummies, I think has to do with America's sweet tooth. We love sweet things. We love sour things. I think when someone's like sitting behind their desk and they can't take a hit of a vape pen, they can just take a gummy. The thing I always tell people about gummies too is you're tempted to chew it and eat it like you would any normal food.
Right.
But the secret to gummies is to keep it in your mouth and suck on it like a cough drop so it gets absorbed into your oral mucosa and doesn't go down into your gut. Because if it goes down into the gut, you may not feel anything. So you'll take another one. You may not feel anything. Take 5 or 6 gummies, and all of a sudden you're lit. And then that's because it's gone through digestion and then oxidation in the liver, and now you've made 11-hydroxy and norcarboxy. Well, guess what? Those things don't go through the blood-brain barrier, and they're going to be circulating all throughout your body. And that's the guy who got the body high from eating too many edibles and had to take the next day off work, because the cannabinoids are acting on the peripheral CB2 endocannabinoid system. It's not getting into CB1. It's really just acting all throughout your body. But I find it really interesting if you look at new markets where edibles are allowed. Edibles immediately shoots up because people like gummies, and I think they like beverages and soda pops and things like that. And now you could put your cannabis in there and you could drink your cannabinoids. But there's no substitution for just consuming flower and concentrates, the way that we do.
I agree. I understand a lot of these products get developed for both convenience and novelty's sake. It's sort of like we don't necessarily need cannabinoids and everything, but we're going to put it in everything because people find it interesting and they'll try it. But I agree. It sometimes the infused products overcomplicate things a bit. One, by introducing variables like sugar and other things. But I wanted to. So I've got about 15 minutes left. I wanted to loop back around to start to cap this off. We talked kind of about the history of the research all the way into the 90s. Let's talk about more modern research. And I think one thing that people hear some of us scientists say a lot is that Raphael Mechoulam deserved the Nobel Prize. And when he passed away, a lot of us on social media voiced our frustration that he had not received the Nobel Prize. And so let's talk a little bit about that for folks that are kind of on the outside there and don't understand why folks are making those claims. Why would you say Rafi deserved the Nobel Prize?
Well, Nobel Prize is a very politically charged award. There's a lot of politics involved that deal with not only the United States but international politics, because it comes out of Sweden. And the committee is over there in Sweden, and it consists of international scientists. And on our Hash Church episode that we had dedicated to Rafi, actually we renamed it. We call it Hash Temple.
Nice,
now,
nice.
since Raphael was a practicing Jew, we figured, well, let's call Hash Church Hash Temple, since this was dedicated to him. So on that episode, Ethan Russo talked a little bit about the previous efforts to nominate Rafi for the Nobel Prize. So Nobel Prize really has to do with not just a really important discovery, but it has to do with the impact that discovery has in the field.
Yeah.
And if you look at it, I mean, the discovery, well, let's back up for a second. His structural elucidation of THC was extremely important scientifically and extremely important in the chemical literature, but that wasn't really worthy of a Nobel Prize because, other people had isolated it first and other people had talked about its structure, but then Rafi kind of came in and cleaned up all the previous work.
Right.
But what certainly was notable was his work that they did after the discovery of THC, and after they found out that it was the principal active component, so-called toxic component, or the thing that led to intoxication from cannabis, was how does it work? And is there something here that is more generally applied to medicinal drug discovery than just smoking cannabis and feeling the effects of agonizing one's TV or one's CB1 receptor. So here's the joke for your next party, Jason. When you want to ask somebody, hey, do you want to get high? Rather than saying that, it's like, let's go agonize
You're
some
right.
CB1 receptors.
Yeah.
Or I liked when they found out, this was recent work within the last 10 years, that actually cannabinoids are involved in neurogenesis.
Mm-hmm.
So when you told your roommate in college, hey, let's go kill some brain cells, you're actually doing just the opposite. You
We'll
were growing
stimulate
brain
some new
cells.
ones.
Right.
Yeah.
But I think the real interesting aspect about the, where this brings us today, and you asked about like the ongoing research that's being done. So we're finding more and more out about how these things work in the body. And that, to me, if you look at that body of literature, doesn't happen until Raphael's seminal work in collaboration with Professor Howlett
Yeah.
and William Devane and Pertwee and Roger Pertwee in Scotland. The discovery of the endocannabinoid system is worthy of a Nobel Prize. There's no question about it because of the impact that it's had in the industry. When they discovered the receptors and then later on the ligands for those receptors, there is not one pharmaceutical company on planet Earth that did not pay attention to that and immediately started developing drugs to act on those receptors, right? That's how we came up with the spice compound, the JW compounds, the WIN compound, all of the cannabinoids that are in clinical trials right now that are not phytocannabinoids all came out of Raphael's discovery of the endocannabinoid system, which was really the missing piece. We knew that humans love this plant. We know that it got them happy or high or euphoric, but they didn't know why. And it was that seminal discovery of the CB1, CB2 receptors of anandamide and 3-AG that is certainly worthy of a Nobel Prize. And so what Russo was saying is that it was a couple of years ago, I guess, that this was kind of petitioned up to the Nobel Committee. And, there's a lot of politics involved. And certainly here, the politics at play is that cannabis is illegal. It's against the United Nations treaty to be involved in cannabis. And so I think there was a lot of knee-jerk reactions on
typical,
just not focused on anandamide and CB1, but focused on that bad plant cannabis, which is part of the whole war on drugs.
Yeah.
So I think if he would have been alive for another handful of years, he could have certainly been in the running to get it. They don't award a Nobel posthumously, but you can imagine that, Alan Howlett is still alive. William Devane, unfortunately, passed away a couple of years ago. Roger Pertwee is still alive. So some of Mechoulam's co-workers who were responsible for the discovery and exploitation of the endocannabinoid system could reasonably get the Nobel Prize. I'm sure that they would add Rafi posthumously to that Nobel Prize, but I think that it would be really interesting to see whether or not that has legs or not. Because when you look at some of the Nobel Prizes, you say, okay, I guess that was important. I don't really understand the chemistry, but I guess it was super important.
Yeah.
But when you
this
look
one
at
is
the
so
overall,
obviously important.
I think so.
Yeah.
But tainted with the political stripe that cannabis is always painted with, which is, oh, you guys are, at a Cheech and Chong movie or seeing a Grateful Dead concert and all you want to do is get high, right? No, I think that there's really a place for herbal medicine, and maybe that's the most important lesson. Cannabis teaches us a lot of lessons, right?
Yeah.
It teaches me every day. I learn new things about cannabis every day. And I think the main lesson is that maybe the monomolecular medicine model of the FDA isn't entirely correct. Has the FDA approved life-saving medications?
Right.
Absolutely. Are people alive today because of FDA approved drugs? Yes, definitely. But is there a place for herbal medicines
Right.
like cannabis and like echinacea and
Yeah.
like other herbs that have been used? Chinese medicine. I tell you what, I had a condition. Now, I won't tell you exactly what condition it was, but I was in a lot of pain. And I went to my friend who has a Chinese herbalist who whipped up a little
Mm-hmm.
concoction. And I can tell you that got rid of my pain immediately. Immediately. So there is something to
Mm-hmm.
herbal
Absolutely.
medication. And maybe that's the most important discovery here is that herbal medication has its place
Yeah.
in the modern pharmacopoeia. It's not going to replace prescription drugs. It's not going to save your life if you're dying from certain conditions.
Right.
But certainly, medical cannabis, and all cannabis use is medical because we know about its antioxidant action. We know about how it acts on the body. So all cannabis use is indeed medical. I think what we need to do is we need to be able to, you know, have both worlds. FDA
Yeah.
approved
Yeah.
drugs with single
Mm-hmm.
active ingredients that are targeted for certain diseases, but obviously this medicinal herbs that we can grow ourselves. I mean,
Yes.
my
Yeah. Make
God,
our own
imagine that.
medicine.
Do you think the pharmaceutical companies want us to be able to grow our own medicine?
I'm sure there have been some calculations done of um, what the market dig might be if people stop taking aspirin as much or Tylenol as much. Some of these, I'm thinking more of like the over-the-counter, very, very common day-to-day medicines that can actually be easily replaced by medicinal plants.
Well, Jason, you probably know this because I think we discussed this in our podcast. Um, uh, Acetaminophen,
Right,
which is
right.
Tylenol,
Yep.
undergoes a hydrolysis in the body, and then it conjugates to arachidonic acid to make a cannabinoid called AM404, which is responsible for the analgesic activity. So basically, Tylenol
Yep.
is a pro-cannabinoid.
And it's
It turns
it's
into a
and it,
cannabinoid in the body.
And it's worth mentioning too that so many pharmaceutical drugs, and acetaminophen being one of them, it was developed and approved before we knew exactly what it did or how it did what it
That's
did.
right.
And it's
That's right.
and it's important to understand that because when it comes to cannabis, there's often this double standard applied where folks will say, well, no, 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 we don't know how it works yet. We can't, we don't know enough. We need to do more research. And it's like, but a lot of these other drugs, a lot of over-the-counter drugs, which are known to be very toxic on your liver if you're taking it more than sparingly. A lot of these different things, we know that they're more dangerous than herbal cannabis, and we didn't know how they worked, and they got approved. So
I mean,
it's
alcohol and cigarettes, you don't have to look any farther than those two
yeah,
things right there.
yeah,
Right.
absolutely,
And when you look at prescription drugs and the way that the opiate crisis has kind of
yeah,
fleshed
yep,
itself out here, you'd say that there are bigger things that are much more dangerous than cannabis, and the fact that opiates and all these other things are toxic, and cannabis is not toxic at all. I'm talking about toxic that
right,
you
right.
can die. I think the only way you can die from cannabis is if you break the law and get shot by a cop or something, right?
Absolutely. Yeah. Yeah. Yep. And it's really ridiculous when you do zoom out and you see like so the story that we've told today going back to the 1800s of the roots of some of the stuff to understand like what is cannabis doing and why, and to see all of this development and what it's led to in terms of the 2000s in general, this um this particularly this century that we're in, so the first 100 years of the 2000s, I think we're gonna look back on this as a huge, huge
Yeah.
time of cannabinoid therapeutic development. It's already happening. Every pharmaceutical company
Hmm.
is developing cannabinoid medicines in some way. And most of this is public. You can go on their websites,
Right.
look at their development timelines, and you will almost certainly find cannabinoids in some form or another being developed. The understanding of the endocannabinoid system, and then from that, the endocannabinoidome, it changes the way we think about physiology, the way we think about medicine, and those effects are going to cascade for a very, very long time and lead to very interesting new questions about physiology and new discoveries. Like we were saying, there are certainly other systems. There's still many orphaned G protein coupled receptors that we're still wrestling
That's
with
right.
to understand, what is
We
in
don't
the.
know what they do.
Exactly. Yeah, we don't know.
And we don't know how they interact as a system. I mean, we only know like 10% of the instruments in the orchestra, yet
Yeah,
there's this
exactly.
full orchestra of instruments, and we think that just the 10% that we know is all there is, when there's a whole iceberg of stuff underneath the surface that we don't even know exists yet.
Well, and maybe some of the Rafi's contributions is his gift to us is because part of his gift is not just what he's done, but his writings and the fact that he has
Yeah,
told the story. That the gift is being able to see the frontier, to be able
right.
to see where things are headed, where they're going, where the mystery lies. He took great care to help ensure that the story is maintained. And I really recommend, if anyone doesn't read anything else, Rafi's a great chapter in the book Cannabinoids, which is now starting to be an outdated textbook on cannabinoid science. But I think the last updated edition was in 2014 or something. But he has a chapter in there. I'm trying to remember. I might have it right here. It's the very first chapter of the 2014 edition, where he just talks about everything we've discussed today, just the general history of cannabinoid science and where it's headed. And I really recommend folks, if you have a way to look that up and read it, it's like Mark said, when you read his writing, you can really get a sense of where his head was at, his general passion for the project that he's been working on. Like he was aware of his place in the story, and he was
He was.
aware that the story continued after him, and he did everything he could to try to like lay these kind of breadcrumbs around to help
Right.
people see that frontier and understand that they have a place in it as well. So I don't know. That's something that I've kind of gotten out of our discussion today is, it's a real gift to be able to see the frontier. And that's largely because of both his work and his attention to telling the story and helping to prepare the future generations of scientists.
Well, I think what he says at the end of the movie, The Scientist, to me is absolutely just fascinating and spot on, just exactly where this thing should go, which is that, there's 8 billion people on the planet. No two people are the same,
Right.
right? And each individual person, each individual human reacts differently in different situations, right?
Yeah.
And that the way that we react and the mood that we get in, and how we think about things, is this manifestation of the endocannabinoid system, and how, like, when my daughter comes back, hey dad, I'm on the dean's list, and I
Right.
feel that bliss, my body must be making anandamide or God knows what other molecules that are
Right.
acting on similar sets of receptors. But our emotions and the way we react and the way we perceive things is this chemical communication signaling, that maybe the endocannabinoid system is just one component of
Right.
that, and there's 10 other things left to discover. But he posed to a mathematician. He said, well, there's 8 billion people on the planet. Let's see, there's these 5 molecules, there's these 10 receptors. Could this actually lead to the diversity that we have in the human race?
Right. Right.
And different personality types, and if you look at your personality versus my personality, in some way it's an expression of emotion, and
Mm-hmm.
those that emotion comes deep down inside from, compounds acting on receptors. And smell particularly is like one of those receptors that as soon as you smell something, you'll remember back. Like when I
Right,
smell
right.
turkey dinner, I think of my grandma's house at Thanksgiving, right? So the certain smell that you associate with certain things, I mean, to me, there's a much larger thing going on
Yeah.
in the human brain, and the endocannabinoid system is kind of like is showing us the whole, saying, "Hey, man, on
Yeah.
the other side of this," and how these things are all connected. I do believe, yeah, his lasting impact will be how he went from the structural elucidation of THC to understanding the receptors it acts on, to understanding the natural ligand
Yeah.
that the body makes that mimics THC, and that these systems might be duplicated
Yeah.
throughout the body, and that there could be interaction and interplay between the two. It's really a combinatorial mathematics problem that could certainly result in the number of different personalities that we have in all 8 billion humans that walk on Earth. So yeah, it's a fascinating thing to think about too, what he mentions, which is that in the procreation of life itself,
Yeah.
you have a lowering of the immune response when the woman gets pregnant with the
Mm-hmm.
father's sperm. There should be a complete rejection of that genetic material, but there's not.
Mm-hmm.
And obviously this must work because there's 8 billion
Right.
people
It's
on the
they keep
planet.
being
So
born.
it works.
Yeah.
But wouldn't it be great if somehow the discoveries of the endocannabinoid system we start figuring out a lot of these things, and what ends up happening is that cannabis, which again was the thing that led us here, isn't where we end. Like Rafi
Yes,
says,
I agree. Yep.
you know how you start research, but you never know how it's going to end.
Yes. That's a
Just like this
really
podcast.
that's a really great way to, I think, cap things off. And I'm really glad you shared that too, because that's actually my only connection directly to Raphael Mechoulam was through email. We talked through email, and I was very grateful for those interactions. I totally understand what you mean, where it's like an email from Raphael Mechoulam shows up in your inbox, and it's like, what? Wait a minute. And
Makes your day.
and one of the last things that he communicated to me was exactly this: his thoughts on the intersection between the endocannabinoid system and personality and subjective experience.
Yes.
And that's really, his head was really there. I talked to, that was in 2020 or so, that we had that conversation.
Oh man.
And
Yeah,
so
we're gonna miss not having him on planet Earth. I tell you what, he's it was in our Facebook cannabis chemistry group. He joined a few years back, and every once in a while he would put a comment out there,
yeah,
and it's like, oh my God, Raphael's on Facebook. It's like, but I think he was in it because he really loved. And I think that's one of the reasons why I think he's kind of like, he was happy about me because he saw me as being someone maybe like himself who was a young chemist who
hmm,
wanted to learn more about these things and realized that, you can't do it all, and there's no one scientist that knows
that's right.
everything.
Yeah,
We're standing on each other's shoulders.
yeah.
And just as he was standing on the shoulders of Adams and Woodspivy and Easterfield, I'm standing on Rafi's shoulders, and he hasn't pushed me off yet, so I'll just continue standing there. But I think, Jason, what you're doing with Curious About Cannabis, I mentioned this to you last time, it's important to make sure that we get the facts right because
Yeah.
there's a lot of misinformation. I deal with it on a daily basis. Misinformed people who are blogging, just completely bullshit. And so
Yeah.
it's
Yeah.
important to make sure that we get certain things right. And that's why I wanted to do this with you today, just to make sure that everybody knows that, he wasn't the first to isolate
Yeah.
THC. He wasn't the first to draw the structure of THC, but he was the first to elucidate it from natural material. And
Yep.
that's an extremely important contribution that led to all of his work after that.
Absolutely, and it's also important to note that anytime, because I've gotten pushback before when people report, because like some pretty big like news articles will report that he discovered THC, and I was like, no, he didn't discover THC, and that's not a dig at him, and like he would want you to know that too,
Right.
like
Oh,
he
yeah.
wanted people to understand the contributions of the scientists before him, and so I hope that that's clear too, like this all of this sort of clarification and everything comes not in any way to reduce what Raphael Mechoulam
No,
did, but to help people see where he fit
that's right.
in the story, and the fact that if he were here, he would tell you that. He would remind you that about all of these researchers that came before, and that
Right.
he just picked up the puzzle and kept putting pieces together.
The important characteristic there, Jason, is something called humility.
Yes, yeah,
And it's one of the most endearing qualities that any human could have, is to know that it's not about you. It's about the greater thing. And yeah, you take yourself out of it. And I think he had a lot of humility, and I
he
think
did.
that I do too as well.
Try every day.
Yeah,
Well,
that's right.
this has been great. I hope that everyone listening, I hope this has given you a little bit of insight into, not just Raphael Mechoulam's work and contributions, but sort of this bigger story of cannabinoid science. And then we also spun out into some really important discussions that are relevant to the cannabis industry today, and got into some good technical chemistry too. So this has been a good conversation as always. So thanks.
Hey, Jason, remind them of our deep dive. So if you
Yes.
folks are interested in learning more about the pre-Mechoulam era of cannabis chemistry, I think Jason's been kind enough to put the deep dive up on YouTube. So,
Yes, yeah, and like I said, it's like 3 hours long. It's really in depth, and we still need to get together to do the one on terpenes too. We wanted to go
oh
through
yeah, we're gonna do,
and do a deep
yes,
dive on terpene chemistry. We'll do that
yes.
sometime a little later in the year. That'll be a really good one too. So yeah, go find that. It's available for free on YouTube. I think it's on the podcast as well, so it should be on the streaming platforms as well. Although the video I recommend, because there's so many visual aids in that
That's right.
presentation, that I really particularly recommend the YouTube, so that you can see the papers, because through the whole presentation, you can actually see the references to the quotes and everything. It's just a lot really helpful to digest it all. So yeah, check
I
that
went back
out.
and watched it myself.
Yeah, I did too, actually, not too long ago.
I
Because
think
it reminds me
I
of some
learned
stuff.
something.
There's so much information, and it's easy to get lost in it all. So yeah, so
That's
those
right.
of you that really care about the story and want to understand it, from here, go check out that. There's also a presentation on YouTube that I gave to Southern Oregon University a couple years back that is sort of in the same vein. It goes through some of the history of cannabinoid science, but a little more zoomed out, not so much focused on chemistry, but kind of just the research as a whole. You might like that too. I always tell people, if you want to know the history of cannabinoid science, go look at the deep dive that Mark did that's 3 hours long, and go look at the little hour and a half long presentation that I did. And between those two, you get about the best full rounded view of the last 150, 200 years of cannabis research that you could hope for.
Yeah, and if you have any questions, just put them down in the chat, maybe, and I could periodically check in to see if there's
Yeah, send those
any.
our way. We also have the Curious About Cannabis Discord, so if any of you are on there or join on there, you can shoot me questions on there, and I can shoot them over to Mark as well. There's
Yeah,
all sorts
Jason,
of
if you
ways
get
we can
any
do that.
chemistry questions, feel free to send them my way.
Yeah, yeah, totally. All right, everybody. With that, Mark, thanks so much for being here. Thanks so much, everybody that tuned in, for listening. And thanks so much to Raphael Mechoulam for everything he did to get us here. I wouldn't be here today doing what I'm doing right now if it weren't for his work. So
Neither would I.
much appreciation. And with that, everybody, stay curious and take it easy. Bye-bye. If you like this video, be sure to subscribe, hit the bell icon so you get notified of more videos, and go ahead and check out another video while you're at it.